So I'm going to share my screen and to show the main points of the discussion about the inherited disease breakup session. Hold on. Let me just share this. We had a small group, but we basically identified some diseases, some common interests in different types of cancer. Autism, we had someone from Argentina who's interested in metabolic diseases and all these others. Something we didn't talk about was healthy controls, which we have access to, and given the genetic diversity of the region and the missing diversity in genomic data in general, it could be interesting. So Mexico and Brazil have platforms for single-cell-seq, the 10X. And people talked about, everybody's working on humans using organoids, particularly brain. We have IPS, tumors, spheroids, and PBMCs from control populations. And we couldn't really set on specific questions. And this was a first discussion group. The challenges we identified. Funding. Funding is a big challenge. And the other thing is bureaucracy. We all in the area, even when we have fundings, getting reagents and getting things done and equipments installed and support it takes a while. So the pace in which science and technology evolves, new technologies evolve in the region is slow. So this is something we worry about. In terms of the other challenge, and I think it's a challenge worldwide, is to have enough bioinformaticians and people trained to extract knowledge from all of this data, and single cell technologists. This is something that is new, and I think it's even more challenging. Bioinformaticians we have, we just need training to deal with the single cell data. Tissue banks around the region, we didn't talk much about that, although people have samples, and for instance in Brazil we have a national bank of iPSCs from different diseases, and if we can identify some common research interest, we can set up shared resources. And this is something that we think is really important, which is finding a shared research interest, the flagship project for the region, which would make sense for some specific problems, specific or more urgent in the region, that at the same time we can contribute with new knowledge and addresses needs from the region. And with this flagship project, I think it will be easier to gather people together from the field and to go after significant fundings. And we all agreed there that translational biomedical research is hard to put together, but it's still easier than basic science research in terms of local funding. And there is this difficulty to get good samples. The opportunities, on the other hand, are the unexplored genetic diversity in the area. And I think this is something that we can contribute to the whole field. We also may have unique disease burdens, and mostly infectious diseases, in terms of inherited diseases, we're not sure, but definitely in terms of infectious diseases. The other opportunities would be international grants. And in Brazil, the Ministry of Health has launched in August a national program on genomics and precision health, and this may represent a local opportunity for funding. But that will depend on the economy and the president. So in terms of goal settings, I think the biggest goal is to find the flagship project. And everybody thought that HCA Latin America looks good in the future. It's promising, although there was some concern on how to keep up with the development internationally because of all the challenges we have. We think it's important to have training courses within the region to create more human resources to work with single cell data, generating and analyzing. And we think it's important to have annual meetings and establish committees. And I guess that's it. I don't know, Shim, do you want to add anything else?
I think you covered it very well, Ligia. I think some people may have noticed we strayed from our brief of inherited disease. Hope we'll be forgiven for that. The discussion was broader.
Yes. So I think this is it. This is what we had to present.
So shall I go next?
Yes.
So I was not as organized as Ligia, so I don't have slides to prepare. I won't share my screen, but I can just summarize what came out of our breakout group. It is if I can find my notes. So we covered many of the same opportunities and challenges that the first breakout group did. So I won't really go through all of those. Certainly funding came up, of course, in our group, as I imagine it did in many groups. But we really focused mostly on the idea about how to drive new collaborations and networking within Latin America and even beyond. And also a little bit about training too. So I'll come back to that at the very end. So the main question though is really how do you drive collaborations? And this idea of flagship projects really came up in our group as well. In our case, because we were the infectious disease group, that's naturally what we focused on, particularly with regard to neglected diseases. We had, which included examples such as Chagas disease, leishmaniasis, perhaps tuberculosis, HPV came up as well. I think one encouraging aspect is that COVID has really provided a model for and some experimentation about how do you really drive collaborations and consortia around particular diseases. So that includes things like collaborative publications and meta-analyses, new networks for data and sample sharing, registries of researchers who are doing single cell analysis on particular diseases and so forth. We also felt that disease-focused consortia could be a good opportunity for funding proposals, but also that the inverse is true too, that developing proposals for funding is also a really good way of driving consortia development as well. So it can work in both directions. The point was made that many of the diseases that are a particular problem in Latin America are also a problem in parts of Africa and Asia. So this can drive intercontinental collaborations and consortia as well. And that funders may be particularly interested in this idea. We just heard about this too. Funders such as Gates could be interested in consortia around particular diseases. And then in addition, there are links to particular organs as well. And so, for example, in the cases of leishmaniasis or yellow fever, there's very heavy liver involvement, obviously, and then that could actually provide a natural link to some of the HCA biological networks that are being created right now, including the liver network. So we had a lot of discussion about how you really form these groups. It could be sort of, I mean, obviously there are many tools, including WhatsApp, for keeping groups like these going and facilitating communications. But in addition to sort of disease-focused groups, we also really had a lot of conversation around the idea of studying immune cells as a sort of a cross-cutting way to bring different research on different diseases together under one umbrella, and that in and of itself could be a sort of flagship project. How do what's sort of the immune response, and how do immune cells change in the course of different diseases? So we could imagine that a funder might, such as Gates, might be willing to open up a call for projects in this area, perhaps involving say $200,000 in seed money for proposals for exploratory concepts that involve cross-country consortia. And all of this led us to a concrete idea for a next step, which is to write a review about what we'll call it a human disease atlas could actually look like. What would be the components of such an atlas? What problems and opportunities are there that could be studied? And this actually, we actually got some way into standing this up. So Lucio offered to take the lead on putting together a team to produce an outline for such a review that could then be pitched to different journals. And if we can get interest, then a review on what a disease atlas might look like could then lead to the atlas itself. It could really be the catalyst for drafting the atlas itself. So that was a real positive. I think, and not to, I think Lucio can talk about this more, but I think we would definitely invite interest in such a project. We had a little bit more conversation about other areas as well, some conversations about how do we create networks of computational scientists and bioinformaticists in Latin America. So I think that could really be a goal for HCA Latin America. And then getting back to the idea of online training courses, as I understand it, there are 4 machines now in Latin America, 2 in Brazil, 1 in Mexico, and 1 in Chile. So we see an opportunity for an online training course where we could actually bring presenters together from those locations to do a sort of an online training webinar about, or a multi-part online course about how to use that technology. So I'll stop there. Lucio, anything to add?
Yes, thank you, John. You cover well. I mean, so I was a very good discussion that you have with this group. You saw a great opportunity. I mean, bring this technology, bring this approach of single cell to put light in disease that's extremely complicated for us to understand, like leishmaniasis, Chagas disease, so many other viruses. So I think this approach that we want to do with the Human Disease Atlas can be very helpful for other regions in the world. You know, like leishmaniasis is a big problem in India as well. It's a big problem in North of Africa, and so I think as I see if you can show that you can work in a very organized way as a network. I mean, obeying the very good protocols and good practices. I don't expect you to get problems to get funding for that. So I'm extremely positive that you go to. This can help a lot to solidify solidify the Latin America network. Thank you, John. Now I say Patricia Severino and Christine Wells, you talk about cancer stem cells.
Yeah, thank you, Lucio. Let me see how I share this. We took some notes. Actually, with great help of one of the participants, which was Daniela Robles. She really helped us out with that. Can you see that?
Yes.
Yeah, okay. So we had a great discussion. I think we were a big group, and I think that everybody got to participate, and we got to understand what everybody has to offer and what everybody's willing to do. So here is a summary of what we talked about. So we were able to see that there were lots of common interests within this group. And we had people from different South American countries, which was great. We had Chile, and we had Mexico and for us in Latin America, Mexico is a very big partner, and they are very, they already have results, they already have infrastructure too, so that was great. So a lot of potential for collaboration with groups that are already using the technologies. So Mexico could be a very good partner. And yes, in Brazil, we also have 3 machines installed, and a 4th one coming for the group of Mari Boroni, who got the CZI project. So that's also very important. We also saw that different countries have a lot of experience and a lot of people working with data analysis. I think we have this in common with other groups that also mentioned that. So we detected that as something which is really a very important point, and we can already start from that. We also had several groups with access to biobanks, which provide a lot of diversity for studies that are going to try to understand genetic diversity. So several cohorts, also disease cohorts. We also found out that a lot of groups have access to that. There are a lot of international collaborations that already exist. So specific, the group in Chile is already part of ENBO. So we thought that was really great. They are putting together already workshops for data analysis, which we also thought was really great. Lots of infrastructure already available in Brazil, but also in Mexico, and the genetically diverse cohorts. Challenges, well, funding, it has been already said, is limited in many countries. Biobanks are great, but Jay, he pointed out how SNET frozen samples, we're going to have some challenges. Specific protocols are going to have to be used, and specifically nucleus sequencing or other kinds of technologies just that we have to be careful with that. So spatial biology, of course. Yeah, but we have, since we have all this biobank groups with access to patients and biobanks, there's a lot of sample collection that can be carried out prospectively. We will try to understand how these protocols are working for us and for the specific disease that we decide to work with. And what we tried to do was to set some goals, so some immediate goals and 1-year goals. So for immediate goals, we were able to define a few things. So formation of small hubs to pursue specific projects. Within this group, we could see that that is possible. So we will be in touch. Immediate goals, choose some samples that could be used for benchmarking techniques. Since we have the equipment, we have the expertise from the data analysis groups. Maybe we can use some common questions to set up this benchmarks for us and compare them with HCA so we can see how good we are doing. Create working groups, in bioinformatics, specifically to learn how to work with HCA data, which is plenty available, and learning and discussing protocols for the biology. Meetings with funders. I'm going to talk a little bit more also about this later, but we think that there are many funders who could be interested, like other groups also mentioned. It would be important to understand what their funding agenda looks like and how we could use the idea of equity that permeates HCA and discuss how we could really participate as a group and contribute to the HCA. So the Wellcome, the NIH, the CZI, the Brazilian FAPIs, which can provide funding for collaboration in international studies. Even though they only fund Brazil, but they can get in touch with the other funding agencies and really put together some very nice ideas of international collaborations. And in 1 year, we thought that what we could be doing now is trying to understand what is available in each of the different countries that are contributing or that could be contributing to this initiative. in terms of biobanks, that we could have access to biobanks with people that would be interested in participating, so they could describe what their resources are, specific diseases that we would like to work with, and with that have a clear picture on about how we can really collaborate with the HCA. So besides this immediate small hubs, we think that we can have this more long-term idea after 1 year on how we really stand. And we thought about 3 working groups. One working group for bioinformatics, which could be already working with training students, online courses, as John also mentioned, on how to work with HCA for data analysis, algorithms, and international HCA standards. We think that's something that can already be implemented soon. Working group for meetings with funders, and also, which was something that was pointed out by Vinicius, there are many funding possibilities that we don't really have access to, or we don't come to know about, for example, for fellowships or specific collaborative projects. And if we have a small group of people monitoring those, that could be also great. And a working group that would monitor the progression on establishing this more long-term goals that with which we could tackle genetic diversity and specific diseases. So it would be a group for assessing our resources for international collaborations. That's about it. I don't know, Christine, if you want to add something to what I was saying.
You're on mute, Christine.
Classic Zoom error. I just wanted to say I was really impressed with the energy in the room, and your skills, Patricia, at bringing everyone together for the discussions. Thank you.
Thank you.
There was a lot of common interest in the cancer space, in particular around cancers which are really rare in the rest of the world, but relatively common in parts of Latin America. It was thought that that actually brought a lot of expertise in understanding the tissues associated with those cancers as well. People interested in the genetic drivers, but also very interested in cell-cell communication. And John, the idea of looking at the immune infiltrate in those cancers was a recurring theme. And so I think your idea about looking at the immune cells would be very attractive to this group as well. And I was also impressed that there were people looking at different modalities of measurement, so not just transcriptomics or chromatin structure, but also with expertise in proteomics and metabolomics as well. And that offers some really important perspectives to expand the atlas, particularly in a disease setting.
We have 2 questions here from the participants that were in the groups. Someone is asking about a meeting in 1 year from now, when we would maybe have more to share. I think that it depends, right? Depends on what the world looks like in 1 year. But if we don't meet in person, I'm sure we can meet this way again. I guess everybody agrees with that. Maybe we'll do it in Australia time, right, Christine? Because it was really tough for Christine to be with us here.
It would be great.
Yeah, we're gonna do that. And there's another question here about leveraging funders such as the Gates Foundation to engage with the HCA projects. And we discussed a lot of that. We think that it's about getting together with them and presenting our ideas, what we have, and then hear from them what they would like to be doing and what are their priorities now. So not only the Gates, but the Wellcome, the NIH, the Brazilian funding agencies and others that exist. We believe that if we get together with them, then we can come and find common grounds, right? I think that with that, we should move to the next breakout session 4, Helder.
Yes. Am I on? Can you guys hear me? Okay, so we also had a very nice discussion. There were almost 20 people in that room from Brazil, Chile, Colombia, Argentina, also US, people from CZI. So it was really interesting. The first thing we did was to separate those that were interested in contributing with samples. So they have access to liver samples, brain samples, and people that also have expertise on single cell analysis, you know, so that gave us a sense of what we could do for our small core. Scientifically speaking, inflammation and metabolism is related to infectious disease, to cancer and genetics, everything that's in the other breakout sessions also discussed. So, and of course, as a researcher, we're going to mention the same challenge that you guys mentioned before. Funding, lack of bioinformaticians. So all of these were discussed a lot. And what I will say is just like the regional view, and Musa is going to talk about the global view of what we discussed, okay? So as a regional interest, of course, we do have disease from that are more specifically to Latin America, such as neglected tropical infectious disease, which was discussed in another breakout session, but also in ours. But we also have developing country disease, such as type 2 diabetes, metabolic disease, right? So we had to talk about this, but in the context of the ethnicity and also the socioeconomic status and violence, you know, like how this would somehow affect what we see at single cell level. So what we discussed is how could we put this together in the context of Latin America in terms of like ethnicity and socioeconomic issues. We talk about data integration, so the challenge related to bioinformaticians, the course, the training, everything that the other groups also discuss. But what I would like to mention is the flagship projects that we talked there, because Mariana, which was in another breakout session, Pedro Vieira and Marcelo Mori, they come up with a nice idea for a flagship project that could be a nice start as a result of this breakout session. So for example, they're proposing to focus on creating a macrophage cell atlas, so you could look at macrophage not only in different tissues, but also in different disease. So of course, macrophage has everything to do with inflammation and metabolism. And of course, it plays a role in cancer, infectious disease, everything. So the goal, for example, that as this pilot project would be like to first centralize the single cell RNA-seq data, so we could talk to the standard people from the HCA to really come up with a nice pipeline that we can use to make the data more comparable. So we could get public available data or new data and centralize, but just look at the macrophage. How to define the macrophage population, we will need the help from immunologists and other specialists. So that's why this group actually is asking for help, for people that understand the data analysis, the immunology, and also to create an interface where people can visualize the results, can get extract knowledge, insights from this resource. So for that, we would need computer scientists to help us doing this front end parts to allow other people to look at that. And of course, if everything goes well, we would need people that have a bench lab to validate the results in other tissues and other conditions. So we thought that could be like a nice pilot project that could come out of this, and we could, I hope, show some results in 1 year from now. We also talk about starting some collaborations on ethnicity-related inflammation in tropical disease at single cell level. So we already exchanged the emails, and what I would like to say is that if you want to join us by providing samples or providing expertise on immunology or on computer science, you are free to email us, and then we include you there. Okay, so I'll hand out to Musa now.
Helder, I think you covered everything. I think the parts I can contribute are really kind of rather minor compared to I think you really were quite comprehensive. Listening to what was discussed in, I think, Breakout session 1 around having a flagship project, I think this group had a lot of momentum around what they're calling like a macrophage cell atlas. There's a lot of different people around inflammation biology and macrophages and metabolism. And it's very nucleated around the group of Pedro Vieira, Marcelo Mori, and Mariana Baroni. And so I think this fits well into those ideas. It fits well into funding ideas. We also had a discussion on data sharing, which was quite active, related to COVID-19. And essentially, there was a really nice story told by Jose Ordorvas around how for COVID-19 and for this pandemic, many groups came together to share unpublished single-cell data and jointly authored the publication they had on the nasopharyngeal infection of SARS-CoV-2. And that kind of sets a precedent because I think there are other pandemics that we referred to, including yellow fever and Zika pandemics that have passed through South America in the last couple of years. And those are opportunities for people to do similar data sharing, sample sharing, within of course, the right framework. But this was a issue that was raised. I think in terms of funding, there was a very interesting idea that came up, and that idea was that the Newton funds, which come from the MRC of the UK. So the Newton Fund is a fund that's given to low and middle income countries, and it pairs one British investigator with one investigator in a low and middle income country for 5 years and gives both sides substantial amount of funding. And one of the researchers in the breakout session gave this as a great example of how it's been very catalytic for their lab to transfer a lot of technologies and to build a lot of capacity and to be able to plan for long-term development. And so because we had a couple of people from the CZI in our session, the suggestion was to catalyze paired labs in this way so that you can pair expertise between two different labs in two different geographies, say a lab in Brazil with a lab in North America or in Europe or in Asia or in other parts of the world. And these pairs of labs would have joint projects that they would work on. It's quite different from a seed network project, but what the idea here would be to be able to have very long-term transfer of technology and continuity in this sort of a project. And there was a lot of discussion about that, which I thought was very very positive. I think you mentioned all the ethnicity stuff, Helder, so that was covered. And Helder promised to follow up with all the attendees. We harvested all their email addresses. We created 4 pages of notes that we're not sharing here, but I'll make accessible to everybody. I'll send an email out to Christine. all the people who are participating in the breakout session are also able to edit these notes and add to them. So we can get some follow through on some of the things that were raised. Helder, do you think I've missed anything you want me to cover that I haven't?
No, it was perfect, Musa.
Great. Well done. Good job, Helder.
So I think there's some questions from the audience. Do you want me to read them to you?
Sure.
Okay, just one second here. So we have a question from Shyam. He's asking, if social distancing lasted another year, how could researchers form social and scientific networks? So maybe if one breakout session leader from each group can answer that question. So Musa, do you want to start first, you and Helger?
Well, I mean, I guess, Shyam, you're saying if we have another year of social distancing, I guess we've got another year of Zoom calls, and Zoom stock's going to go up again. So, obviously it makes things complicated if you wanted to have more intimate interactions, but yeah, I wish I could see the future.
Helder, what about you? Do you have any comments on that?
Well, actually, I hope it doesn't last for another year, but we all had to adapt, right? So we do use some tools such as Slack, some things that Trello that helps us to organize the scientific parts. So I'm pretty sure we could do this for the HCA as well, like the HCA related projects. So we could connect people together. For the social network, that it's in addition to Facebook kind of stuff, there is also like some tools that allows you to listen music together and talk, but of course it's not the same. So I hope it does not last for another year.
Thanks, Helder. Yeah, I was in Shyam's breakout session group, and he was talking about the importance of the first year, how HCA Asia came together and how there was just so much energy and ideas bouncing around. And it really wasn't until year 2 when they met in person that things started gelling and coming to fruition. So I think we're just curious to see if the pandemic lasts, how we're going to continue on with our energy and keeping the momentum going. So maybe Christine and Patricia, do you guys have any thoughts or reactions to that? Well, in our group, we had a lot of people working with data analysis, and they don't seem worried yet because they have a lot of stuff to analyze. But in order for us to generate more data and to interact with the groups, of course, we're going to see what will happen. But for us in Latin America, I think we still have a way to go in learning a lot of the techniques and building the groups, building the hubs, building the set of information. Like we said, we want to have access to the data banks, analyze what is possible to be already done, put together people who want to collect samples prospectively. So I think there's a lot that we can still do before we really have to worry about all that. I think we have a lot of, but of course the projects are going to still be going on. Within labs, we can work. I think we just cannot really travel or, like meet together in large groups, but there's so much that can be done. And we in the group also talked about that, how it worked, even though it was in the format of a webinar. So I don't think we have to worry about that. We have to work with what we have. And I think we have a lot to do that can be done in the way that we are. Let's just hope it doesn't last for another year, but we have a lot that can be done.
I just remembered something actually, which is we've been contacted by the Journal of Visual Experiments for this very reason, to publish 2 or 3 technical videos to distribute to people because of it's being used a lot because of the pandemic. People can't travel. So there's a few people in the lab who are making Jove papers, one on, I think, single cell attack, one on doing a technique called CHIRP, which is RNA chromatin interactions. And I guess we're gonna put that out there for people to see in Jove. I think Jove has no paywall, so I think it's free, I'm not sure. So maybe that's a way to,
leverage
transfer technology.
Absolutely.
Yeah, because I think what Patricia just said is important, that like there's still a long way to go in terms of skill set building and things like that. So, these kind of Zoom calls are not going to cut it for that, right? So.
Right. Thanks, Musa. That's great. John and Lucio, do you have any other final comments on how we continue moving forward in the era of COVID?
Yeah, in my case, I didn't stop to work. I come every single day. I work with SARS-CoV-2 inside of the P3. I was out of the bench for 16 years, I'm back working the bench, in the P3. So you need to find a way to deal with that. So you got the TNX machine here 3 days ago. You got the machine installed the next day. You're already looking forward to train people on the technology. And I think you need to find the ways, Latin America, you are the last one to get organized, so we don't have time to waste. We have to move on and find a safe way to move and to make this possible.
Can I add a point, Christine? And that is that having long workshops it takes a lot to organize. People get Zoom exhaustion. It's good for the brainstorming, but it's hard for the relationships. But actually having frequent short meetings by Zoom is fantastic, particularly around with the trainees and early career researchers. Australia is also a very geographically diverse country. We get together weekly for a conversation for half an hour, 2 10-minute talks and a 10-minute discussion, and it's brilliant. And it has built up really fantastic networks. And so I think, embrace the technology, embrace the opportunity that COVID has brought. We're suddenly all familiar with this format that we didn't use before. Let's use it to bring together the diverse countries and the diverse laboratories, even within one country, and just try and find that space to meet often for those nice short windows. Half an hour a week, really easy to find in your diary.
That's a fantastic point, Christine, and I'll use it to highlight and to pitch that we plan to have, I want to say, 4 or 5 brainstorming sessions with the Latin America community and attendees. So we urge you to provide feedback, and in that feedback form on Slido, you can let us know if you're interested in attending those brainstorming sessions, because I think that's where the real work is going to start. That's a great point, Christine. Yeah, Zoom fatigue is a real thing. There was one other question that came through. I will do my best. I think this goes over to Christine and Patricia. You were talking about immune cells. So someone asked, can you expand from microphages to all immune cells?
I think that was Helder who was talking about that.
Helder.
Oh, yes. Actually, when we were talking about the macrophage cell atlas, Norbert, he sent us a link about a project which is already funded by the CZI, which is called an integrated map of the immune system in humans across tissues and ages. So that means that there is already a project ongoing to expand this into more cells, immune cells. And we have to find out what is going to be the overlap between these projects and also the smaller projects focused on macrophage. But to answer the question, yes, there is already a project ongoing for that.
Thanks, Hilder. So I think we are coming up on lunch. Patricia and Lucio, is there anything that you'd like to add before we depart for lunch and dinner and sleep time for those of us in Australia and Asia?
I mean, I think you have a great opportunity, I mean, in these breakout groups, you know, to come with different solutions. I mean, sometimes same solutions, you know, the ideal now is to see all this together, you know, and find ways that you can make this happen now. So I'm very happy with the result. I mean, to see the interaction, the interest of different countries to coming together. And I'm very positive for the future. Patricia?
Yeah, I think it was all great. I think the breakout sessions were very successful. They're also going to be available for people to check them out later on the other breakout sessions that they didn't participate. And I think we all decided to reach out to the people who were in the session so we can have follow-ups on it, on the discussions that we had. And I would like to emphasize what Christine said, that we are planning to have this follow-up meetings next month with people who are interested. And we really think that this is the way to keep the moment going. So, yeah.
So with that, I'll say goodbye, and that will come back at 1:30 p.m. Eastern time. And we'll say goodbye to Cheyenne and Christine, who I think are going to go off to bed now. So thank you, everyone.
Good night, all.
Thank you guys. Thanks so much.
Thank you. Bye.
Thanks, Cheyenne.